Right, sorry, me again sharing the next session, so you can all bear with. So we are now into the genomics session. So we're going to start with PacBio and then follow into 10x Genomics. Paul Gooding, our field application scientist, is up next. He's been with us since 2019. Previously was at AGRF. A few of you will probably recognise him from install and trainings, obviously most recently online. I'm doing like a fun fact about everyone. So yeah, Paul can play the accordion. Yeah, nice little bit of info.
I'm a gentleman, I choose not to.
Yes. But yeah, Paul's going to run us through a range of the PacBio HiFi sequencing applications for plant and animal genomes. So I'll pass it over to you, Paul.
Thanks, Lana, and thanks everyone for being here. I'll share my screen and hopefully we can go from there. Okay, I trust you can all see that. I'm sure you'll all shout if you can't, so all good. So yes, I'm Paul Gooding. I'm FAS with Millennium Science, and so I just want to run through a little bit about the PacBio HiFi sequencing applications for some agronomically important species of plants and animals. But I guess to get into that, first, just a little recap of what is PacBio HiFi sequencing. So we'll just skip through this quickly. So as a reminder, really, you need to sequence your DNA, and that DNA can be any DNA, sort of represented in the yellow and the purple here. So that DNA could be an amplicon, it could be quite short, it could be even sub-kilobase, but obviously longer than that too. It could be, say, cDNA from some RNA, so you could be doing so-called Isoseq, so you're sequencing the transcriptome and the various isoforms. So you've made your cDNA there, and it could be 2, 3, 4 kb, whatever your length of your transcripts are. Or it could be some whole genome sequencing where you've taken the whole genome, you've fragmented the DNA, and again, depending on applications, that might be 10, 15, or even 20 kb or larger. So you've made these fragments and you tidied the ends up, and then you put on a little bit of magic here, which is these smart bell adapters. So these little hairpin adapters that go on the ends of the molecule, and so effectively you end up with what is a structurally linear piece of DNA that you're sequencing, but it's a topologically circular molecule. Okay, and then within the hairpin there's a primer site for the sequencing primer to bind, and that forms a complex with the sequencing polymerase that allows you to do the sequencing. Okay, and the magic here really to get this really long, very highly accurate sequencing, as you can see from the little animation here. The polymerase is able to read, it does the strand displacement, it sequences the positive strand, if you like, around the hairpin, then back in the reverse complement of the negative strand around the hairpin, and round and round and round and round it goes. So it does multiple passes of the same molecule. It is smart sequencing, that single molecule real-time sequencing. So round and round the molecule it goes. So any of these random polymerase errors, represented by the little red dots on the right there, you can screen out, because you can make an intramolecular consensus sequence. So you line up all these passes around the same molecule, you screen out the errors, and you get a very long, very highly accurate piece of sequencing. So that's what HiFi sequencing is. Okay, so that's just a refresher for people as we move into the rest. So what do I want to say about sequencing and assemblies with PacBio? So I thought I'd throw up an example here from rice. Okay, so and just give you an idea of the time scale and how things have changed. So back in 2015, there was a large consortium, multi-lab, multi-country, a lot of dollars, and they decided they wanted to sequence the rice genome. So the rice genome is quite small in plant terms. It's about just under 400 megabases of sequence. So in 2015, they used a back-by-back approach. So they made these back libraries, and they sequenced through using the original PacBio RS2 instrument. And this took a lot of time and a lot of dollars, but they actually got quite a good sequence. It was a lot of manual handling, but they got there and they got a good sequence. A couple of years later, with the newer PacBio Sequel instrument, This was using their continuous long read technology, so it made these same sort of libraries, but it just sequenced really long sequences through. So they were tended to be slightly more noisy long reads. Again, it cut down the money. It did cut down the time considerably, but because they were noisy, they still got into the magic contig M50 of greater than 1. But you know, it was still, they had some noisy sequence. But move on to sort of now, well, 2020, but it's effectively where it is now, really. Moving on to the Sequel 2 instrument, which has got about 8 times the throughput of the Sequel. Again, using the HiFi sequencing now, where you've got this very, very highly long, accurate sequencing. They've really cut down the dollars considerably and the time considerably, where within a few days, and I'll give examples of this as we go through, the speed that you can use and the accuracy to get this massively high, again, contig M50 figure for the rice genome. So that's really how things have improved. So I suppose what I'm trying to say here is that really genome sequencing and assemblies using PacBio data now is really quite routine, actually, and the list is out there if you scan the literature. You know, all the top publications are showing high quality genome assemblies from all sorts of eukaryotic life, really weekly. If you look at the journals, it's there all the time. They're coming out more and more, and there's huge initiatives out there like Darwin Tree of Life, VGP, Earth Bio Genome Project, etc., that are also making it their mission, if you like, to catalogue thousands, in fact, probably hundreds of thousands of species and get these high quality reference genomes, and they're all adopting PacBio sequencing as the core technology for building these genome resources. So just as an example, how far can you push the technology, I guess? Plant genomes, as some of you might know, can be very large and very complex, rice being a little bit of an exception to that. But so PacBio decided that they really wanted to push the boundaries of their HiFi sequencing and what could they really do. And there was a number of reasons. They're based obviously in California, so they chose the California redwood sequoia tree. It's a 9 gig hexaploid plant, so that's 54 gig of DNA content to sequence. Interestingly, it's a sort of locally interesting for them, being in California, of course, but also their long read competitor, Oxford Nanopore, had also tried to sequence this using their long read technology and also some short read technology as well. So it was a bit of a head-to-head. And as you can see, the PacBio on the right there with the green heading, with 22-fold coverage, they got a much bigger genome assembly size than the ONT and short read combined. Better contig assembly there, a better alignment, and therefore a better Busco sign of completeness of the genes. But this is where the data is extraordinary that comes off here, because you've got such long and such accurate reading. You've done this intramolecular consensus sort of on the machine as it's sequencing. So you get very short file sizes, small file sizes that can be easily aligned. It cut down from several months of computing to a few days, which I just think is extraordinary. So in a little bit more detail, I guess, you can see compared to human, the size of the sequoia genome. So it's 9 times the size of the human genome, effectively, with 3 gig diploid, so 6 gig of DNA for us. But really, the Sequoia project that PacBio ran, they were able to collect samples from their favorite tree and do the extraction. They extracted using the Circulomics kit. Actually, if you're interested in PacBio sequencing or making libraries and you're interested in Circulomics, contact somebody from Millennium Science. We'll talk to you about the Circulomics kit. They're very, very good for extracting DNA for PacBio libraries, also cleaning up DNA that you may already have using some of their kits. So do contact us for that. But they made the samples and the libraries in 4 days, ran the sequencing to get the coverage they wanted in a week, and then, as you said, the assembly so quick. So 17 days to do the whole thing from going out into the field to collect to having an assembly, which is just extraordinary. And more to the point, you're ticking these boxes, and often it's put as 3 Cs. We like to think of it as 4 Cs because the compute time is important too. So you've got this highly contiguous sequence, so nearly 2 megabase contigs there with no gaps, high level of completeness, so it's greater than 5x haplogene sizing, and great completeness, nearly 60% Bushko score for completeness. As far as correctness goes, if you looked at the gene sequences, the predicted gene sequences, there was almost no errors in there that made frame shifts in the coding regions. So we know that the correctness was also very, very high. And incredibly importantly, I think that the compute time just so fast, because these sequences are already sort of have been checked, if you like, to make their consensus before you do the assemblies. So really, really fast. So they moved on from this and went back to their favorite tree, got some more needles and extracted RNA as well to do this so-called Isosceles data. Again, very complicated genome, as we've said, with the hexaploid, the 6 components, if you like. And just looking at this, at the bottom, you can see the Isosceles transcripts. So you've got a huge amount of alternative splicing shown in that bright orange there, but also more dramatically, I think, it's just these extremely massive introns that actually these ancient conifers are famed for. Really, they've just got massive introns. Very, very difficult to do anything with this with short read sequencing, but there's some software there that was used called Isosceles, which was able to separate the alleles based on the variants within the coding regions. And what you actually see is it actually pulled out 5 alleles for this particular gene, and that was because any of the variation between 2 of these haplotypes was not in the coding regions. It was only in the non-coding regions where you could pull them apart. So effectively, the software rightly called that there was 5 alleles, but because 2 of them were essentially identical within the coding regions. So this also agreed when you looked at the whole genome sequence data as well, and I suppose combined it gives you more information even about the divergence, say, of these subgenomes within the species, which is also, you know, quite fundamentally interesting if you're into that sort of stuff. And something else that people are very interested in using PacBio HiFi data now is the idea with pangenomes. So there's a big trend really towards this, particularly in plant genomics, for agricultural research and for breeding programs. I guess it's long been realized that to get a complete picture of the genomics of a species, it's not just an individual. You need to find out all the differences with all the different varieties and the different lines, and this is helpful for plant breeding and manipulation, of course. And when you look at the sequence differences between these different individuals, you'll see that there are core genes, and they are obviously critical for the function of that organism, and they're generally highly conserved and they're not evolving particularly quickly. But then there's these so-called dispensable genes, and these are more important there for a lot of environmental stress response, that sort of thing. So they tend to be evolving faster. They're under selective pressure, of course, and they're a major contributor to the phenotypic variation of these agronomically important traits, say within a plant, and they can be used for improving crop productivity potentially, and they probably play a critical role in adaptive evolution and also domestication of some of those species. So if we perhaps consider maize as an example here, when you look at 2 cultivars of maize, they only share about half of their genetic sequences. So it's clear that a single maize variety sequence is a really useful backbone for genomic sort of infrastructure, if you like, but it can't represent the full complement of diversity within the maize species, right? So A lot of these agronomically important traits are tied to structural variants, some of which can be very large. It's a bit like some of the rare diseases we see in humans, but they're really hard to capture these large variants using more traditional short-read next-gen sequencing or say reference database reference type analyses. So a full pangenome of a species, if you like, that captures all the sequence variation within a species would really help to identify a lot of these really useful variants. You could identify them in wild relatives, and they could be integrated into breeding programs with cultivated varieties. You can home in on important traits that don't have, say, SNP information, so you don't have marker information for a marker-assisted breeding. And all of this will go into assisting breeding programs. So another quick example here is in soybean. This is a paper that came out in Cell where they assessed the pangenome of 26 different accessions of soybean. One thing you should know about soybean that they can be very susceptible to iron deficiency. The leaves go yellow, they lose productivity through photosynthesis, so not good for the plant at all. Here, the researchers identified a 1.4 kb indel of an iron transporter gene that has also been implicated using a QTL marker for iron uptake ability. And the presence or absence of this variant made it possible to classify these 26 accessions into two, if you like, distinct haplogroups. One thing you should also probably know that was in the paper is that iron availability to the plant is linked to the soil pH. So if you have a high pH and alkali soil, the iron tends to lock up as an oxide and it's not available for the plant to uptake. But if you have a lower pH acid soils, then there's much more iron available for the plant to absorb. So these haplotypes were split, interestingly, depending on the predominant soil type that these particular accessions were grown in. So this points to a genetic divergence, if you like, for this gene contributing to the soybean adaptation to iron uptake, which is fantastic data to get from the Isosceles data from PacBio. Moving on to an animal, finally. I thought this one might be interesting as well for New Zealand audience with mussels, though this is a Mediterranean mussel variety. But again, they looked at pangenome of this species, I think from 16 different groups, and found massive gene presence absence variation, PAV, presence absence variation within the pangenome, and a lot of this was genes that were a direct response to stress and survival, and it suggests an adaptive role for this pangenome architecture in the mussel. So again, very interesting data to have. There was a special edition in Nature this year where they looked just basically at the vertebrate genome project. So it was a number of publications looking at high quality genomes, all using PacBio sequencing assembly using HiFi. Yeah, there was a number of publications there. So if you're interested in this sort of stuff, look up this particular issue and you'll find yeah, there's a lot of sort of broad biological research stuff going on there across all the vertebrates. And just to sort of finish off, really something a little bit quirky, I suppose, where we are, or where I am anyway. Let's finish off with a monotreme. So there's examples here for looking at the genome of monotremes. So for this platypus and echidna. So monotremes, they have a very unique reproductive strategy, if you like. So they lay eggs, but they also nurse their young once they're hatched. So now it turns out that many birds and insects have multiple copies of the BTG gene, vitellogenin gene, and this is involved in the production of egg yolk. Okay, so fundamentally important for this sort of strategy, right? Mammals don't have this gene, but the the platypus and echidna genomes have shown that they do have a single copy of this rather than multiple copies that birds, for example, have. So it suggests perhaps that this is something that that humans or mammals, including humans, have lost rather than a unique and clever strategy that monotremes have. So again, something fundamental in biology that you can explore through the genome when you've got this kind of very accurate and long read sequencing. And just finally, just to say, obviously you need DNA in order to be able to do this. So until very recently, it was limited by the amount of sample you could get. But now we're sequencing such things as these little metazoans, these little springtails that are shown here. There is an ultra low DNA input method now to go into making PacBio libraries. It does involve some amplification, which none of their other protocols do, but it can be done. There's now effectively sort of 3 ways of going into collecting your sample, if you like, for sequencing. So you've got the standard HiFi sequencing where you need about 5 micrograms of starting material. It's come down. It used to be 15. It's improving all the time. So again, for a lot of samples, we're able to get 5 micrograms of DNA quite easily. There's a low input method that's been more recently developed for these smaller creatures or smaller people, babies and things. So this has been developed by some of the human genome research labs and looking at rare diseases in infants and things. So it's down to 300 nanograms, and so that's very, very useful. Or the ultra low one that I mentioned, where you just need 5 nanograms of DNA, though there is an amplification step in that. But again, it opens up possibilities for things like needle biopsies in humans or very small creatures, you know, legs of spiders or something like that. So so see, these are some of the ways that PacBio sequencing has been used and is moving forward. So I hope that gives an overview, a little bit of what you can do with PacBio. And I think my colleague James will talk next, perhaps with some more relevance to what it might mean for people in New Zealand. So so thanks all, and it's been a great day actually. I've enjoyed the whole session so far. It's been really good. Cheers.
Thanks, Paul. Really great. Is there any questions for Paul?
Oh, I can't possibly have answered everything. Come on.
I must say, every time I look at plant genomics, I remember how lucky I was to do bovine.
Well, I suppose I'm showing my history because I did a sort of plant molecular biology background, so so it's nice to be able to talk about plants because most of the time I don't get to talk about plants, so it was good.
Yeah. Okay, if there's no, just for the interest of time, I might just introduce James then. But if anyone, as always, if anyone has any questions, just pop them up. James is our program director for genomics. He's been with us 13 years. He's a wealth of knowledge on all things genomics. Yeah, well, he can tell you, but his background was in with poppies from memory. Yeah, with plants. Yeah. The plant poppy, yeah, anyway, you know what I mean. Luckily it's the last person I'm introducing because I'm losing my words. And he is very good at origami. That's his little snippet. So yeah, I'll pass it on to you, James. Thank you.
Thanks very much, Elena. Yes, now everyone knows I've got this strange origami habit, or hobby, if you like.
It's great.
Yeah. Anyway, yeah, thanks. What I'm going to do is I'm just going to lead on from Paul, dig in a little bit more about HiFi and talk about technology choices and access for New Zealanders, or access challenges in New Zealand for long read sequencing, or in particular PacBio sequencing. I noticed a few people in the poll mentioned that they were planning to do some type of project, but using some other technology other than HiFi. So I guess the aim at the end of this is to hopefully convince you that maybe maybe you should consider HiFi for your project if it's a fit. And at the end of this, I'm going to go through a few of the technologies and their strengths and weaknesses in for various different applications. So as Paul mentioned, we've talked a lot about HiFi, and we are now hearing a lot of people refer to HiFi as game-changing technology. And this slide came from someone from the US, a researcher in the US, Heng Li, who pointed out that in 2008, Illumina reads came along, short reads, and then about 5 years after that, we had noisy long reads, ONT and PacBio reads, which were somewhat a game changer, allowing a lot of interesting things to be done with single molecule long read sequencing. But now, 5 years later, we now have these PacBio HiFi reads, and they really are turning into they are being referred to as a game-changing technology. And it's in the last, it was probably 2020 that it really sort of took off, and now we're at the end of 2021, and we're seeing the results of that. So as Paul mentioned, these reads are really highly accurate, and everything about PacBio now is HiFi reads. Paul talked about assembly of genomes and how contiguity, completeness, correctness, compute is all important in a genome assembly project, and HiFi reads are perfect for that. Also in human genomics, the extraordinary accuracy of the reads, the even coverage, and the genome completeness gives you excellent allele resolution and long-range phasing. And because they're so accurate, you get best performance for all variant classes, including single nucleotide variants, indels, and structural variants. And PacBio, or some researchers in the US, through this FDA Truth Challenge, compared various different technologies for being able to call variants across the entire genome, the whole range of different variants that we tend to typically talk about, and showed that PacBio HiFi was the most accurate with precision and recall of any of the technologies when they compared against a truth set of a genome that had been completely sequenced to death with everything. So they truly are giving you, like, if you're considering using other technologies, you're compromising. If you're going to do an assembly with ONT, it might look good on the metrics, but you can expect that there's going to be a lot of errors in there. And is that really can you really say you've got an excellent assembly? And a lot of people these days are now thinking, let's do it once and do it properly when it comes, in particular, when it comes to reference genomes. So how much more accurate are PacBio HiFi reads compared to ONT reads? I know that there's a lot of ONT sequencing in New Zealand. It's easy to access. You can get a little sequencer and do some sequencing. So I know there's a lot of people using it, and there's discussion about, it's getting better and better. But I just wanted to show this chart up to show you how it actually compares. When we talk about HiFi, we're talking about Q33, 99.95% accuracy of an individual HiFi read, with one error every 2,000 bases, which is extraordinary. And this is the accuracy of the ONT reads as it stands at the moment. And with the latest release of ONT, they're talking about maybe bringing out something else. But you can see that, they've got a long way to go. And the only way at the moment they're getting the sort of accuracy that PacBio have is to basically clip off all of these reads and just give you these highly accurate ones, which is extraordinarily expensive. So HiFi reads are super accurate, and they are game changing. And Paul mentioned some of the projects that they've been where PacBio is the core technology for making reference genomes, including Earth Biogenome Project, Vertebrate Genome Project, Darwin Tree of Life, and others. Just in the last couple of months, PacBio announced another partnership with the European Reference Genome Atlas Initiative towards the end of September, where PacBio HiFi sequencing is going to underpin that effort to produce a reference genome atlas in Europe. So it's pretty exciting at the moment for PacBio, and this is really driving a lot of interest in the technology, the HiFi reads. I just wanted to mention also, I know Paul mentioned a whole lot of examples, but I wanted to put this up because these are some assemblies that have been done in Australia. This is a Pagona, a lizard assembly, which produced an assembly with a contig N50 of 203 megabase pairs. I mean, that basically means that this genome is virtually assembled telomere to telomere of every chromosome. They've actually looked at one of the contigs, and it had all of these low complexity regions at the ends, at both ends of the contig and in the middle. And it was basically, you look at it and you go, well, that's just one whole chromosome completely assembled. So I think we're at the point now where PacBio can say we can virtually completely assemble genomes with this with just HiFi reads. With some caveats on that for some really low complexity things where you need to do some manual curation. Here's another one, a macadamia assembly, which was virtually completely assembled as well, which is some work that was done in out of University of Queensland. Oh, the previous work I should acknowledge was done, oh, it's at the bottom of the slide, through ANU and Garvan Labs and at the Garvan Institute as a collaborative effort. And this one was done by Robert Henry out of Quaffie, at University of Queensland, where they virtually completely assembled the macadamia genome with each one of these contigs virtually representing a chromosome. Human genome has also been resolved recently, and all of this work that I'm showing you is all this year. And Adam Philippi led an effort to completely assemble the human genome, which and this assembly introduced another something like 100 to 200 megabase pairs of new data into the human genome assembly. So we can now say we've virtually completed the human assembly, and it's all because of HiFi. The only other technology really that was quite useful, and this is where I think ONT, Oxford Nanopore, is quite a useful technology, is being able to resolve these little bubbles in the assembly where you've got really low complex, really long repeat regions like ribosomal DNA tracks, or really super low complexity regions where you need to manually sort of join the contigs together. In human, so leading on from that previous slide, in human genetics, human whole genome sequencing, PacBio has a particular strength in rare disease. So there's a lot of groups now that are finding that, well, we know that there's a lot of unresolved rare and inherited diseases that short read sequencing just hasn't been able to resolve. 50% or more, even up to maybe 3/4 of diseases that we know they're inherited. You throw all of the short read sequencing at them and you can't find the causative gene. So there's a lot of interest now in using PacBio to do comprehensive variant calling across a human genome. And there's quite a lot of collaborations now, all in the last year, that have started, where PacBio is working with various different groups to look at how HiFi sequencing can give comprehensive variant calling to diagnose rare disease. So there's one of the Children's Mercy Kansas City Hospital has a collaboration with PacBio. There's one at the Rady Children's Institute has a collaboration that's announced in June. Just in November, ARIP Laboratories now have a collaboration with PacBio to study rare disease diagnosis, and UCLA Health just in the last, actually 7th of December, what is the date today? I can't remember. It's about 5 days ago. They just announced a new collaboration as well. And of course,
a day ago.
A day ago, is it? Yeah, so yesterday. So I mean, this is hot. Everyone's onto HiFi sequencing now for these types of projects, and and this is why it's just game changing. PacBio also announced middle of this year a collaboration with Invitae or expansion of their collaboration to really drive the throughput of HiFi sequencing, so you could sequence lots and lots of genomes. So it's not just whole genome sequencing that PacBio is useful for. Paul, I think, gave some examples of RNA sequencing and Isoseq. You can always also look at complex populations, microbiome work. Highly accurate long reads are really useful for 16S and also for building meta genome assemblies because of their length and accuracy. And I also talked about variant detection. So there's a number of different applications that PacBio actually have good strength in. And what I thought I'd do is compare some of those applications to some of the other common sequencing technologies. I'm not going to talk about scaffolding technologies too much, as BioNano and HiC as scaffolding technologies for assemblies, and linked reads, which is unfortunately now dead. But I thought I'd just put up a chart, or I'm going to put up a table and compare PacBio to the other technologies of where their relative strengths and weaknesses are, to give you a bit of a snapshot, I guess, of my thoughts around where PacBio is strong. Now, I already talked about large genome assembly of eukaryotes. I know a lot of people use ONT, but for all of the things that we've talked about, PacBio is definitely the gold standard here by quite some margin now with HiFi. If you're considering using Illumina, really just come and talk to us, because I've seen a lot of projects and just not meet the expectations that the researchers had when it comes to assembly of large genomes for eukaryotes. In bacteria, bacterial assemblies with PacBio is relatively trivial now. We can close genomes to completion for pretty much all bacteria, and we can do that in high throughput and get methylation information. Illumina is not too bad at that. Not too bad at assembling bacterial genomes, but you do end up with a lot of contigs, and that's where people usually need to use a hybrid of both ONT and Illumina to do bacterial assemblies, which sort of does work okay, but you need to use two technologies. The pipelines are a little bit trickier, and why would you do that when you can just throw one technology at it and get the result that you want? In population genomics, so I'm talking about large numbers of samples here, PacBio is good. Paul talked about pangenomes. PacBio is perfect for that. But I guess when it comes to the throughput, and a lot of people talk about population genomics, talking about hundreds, 200, several hundred, or even 1,000 samples. The throughput of PacBio to do that number of genomes on one of their systems is where they need to improve, and that's where the collaboration with Invitae is geared at trying to increase the throughput. So that's where a lot of people still, go to short reads for population genomics, because they got the throughput and the cost is a bit lower, and you can do lots and lots of genomes. But they usually augment or add to those projects and underpin them with a few high quality PacBio genomes, so that you have some type of really high quality reference for the population that you're looking at. I talked about comprehensive variant detection with the rare disease diagnosis. PacBio is definitely a top technology when it comes to that, compared to ONT and Illumina. For isoform and splice variant transcriptomics, PacBio is perfect for that. You get the high accuracy and the complete transcript, which is unambiguous. Whereas you can get the whole transcript with ONT, but because of the error rates with ONT, you have sometimes often trouble identifying the actual splice sites and down to the nucleotide level if you've got errors in the data. And short reads, you're basically guessing based on coverage of different exons from a reference databases to trying to understand how the splicing occurs. So with gene expression studies, quantitative gene expression studies where you're wanting to find out up-down gene expression, PacBio just doesn't have the throughput yet, but watch this space, it's coming. There's some new protocols that are being released by PacBio to really boost the throughput on the existing systems. ONT, again, doesn't quite have the throughput, but it's higher than PacBio for transcriptomics at this current point in time. So most people do sort of default to short reads and for their quantitative gene expression studies. Microbiome ID, 16S, PacBio has a real strength in that space because of the high accuracy. ONT, I put a few ticks here because ONT is good for transporting things and going to on site somewhere where you got trouble with, maybe you want to get a fast result, or you don't want to carry a lot of samples. You can go on site and get some interesting data with ONT, but it's not going to be to the level of PacBio's accuracy. In metagenomics, I haven't seen really any high quality ONT metagenomic assemblies. It's usually some sort of combination with short reads and Illumina, whereas PacBio is really quite good for that. A lot of people still fall to Illumina though, because the throughput again is higher for metagenomics, and you do need more sequencing. The last one I'm going to put up here is SARS-CoV-2 sequencing, and PacBio have just released a new protocol or new assay for SARS-CoV-2 sequencing, where they use molecular inversion probes rather than amplification-based strategy, which allows them to get around any problems that you might have of detecting new variants of SARS-CoV-2. So with the type of probe technology that PacBio is using, it is nowhere near as prone to problems with coverage because of new variants popping up. All of the short read technology that's currently used for sequencing SARS-CoV-2 genomes relies on amplification of the genome using a set of primers that are designed to the genome in, I guess, more conserved spots to amplify up a whole panel of amplicons and then sequence them. And you do get dropouts. There's multiple versions of these panels that are being produced, and I'm sure that an Omicron panel will come out soon. But you've got to question whether the next variant that comes along, which we know will come along, whether you're going to need to build another panel. So this is where PacBio has a real strength in their probe-based technology that they've released for their assay, and also the long reads that allows phasing and understanding of quasi-species within an infection as well. So you can see real time the variation that's happening. So here you can see, like, PacBio does have a very strong strength in a number of these applications, in particular large genome assembly, comprehensive variant detection, isoform splice variation, the microbiome ID, and virology. So if you're considering any of these projects, I would encourage you to come and talk to us, and so we can dig in a bit more about how you can use HiFi game-changing sequencing for your project. So now I'm going to flip to, I guess, New Zealand. This is my last slide, and how can you access PacBio in New Zealand? What are the challenges? So currently, the number of installations of PacBio sequences in New Zealand equals 0. We would dearly love to change that. Definitely come and talk to us if you're interested in being part of a collaboration or a initiative to bring a system into New Zealand. I'm already talking to a number of people about that, and I think it's only a matter of time because of the utility of the PacBio as to what you can do with it. So access is currently through international service providers or international collaborations. There's a number of people in New Zealand I'm aware have international collaborations, and they access PacBio sequencing through those. But if they don't, then it's basically get some quotes from international service providers and see if you can get space on an instrument. Many New Zealand projects, they're working with native species and working with these types of samples is culturally sensitive in New Zealand. It requires respect of the cultural expectations and to maintain control of and return samples and data. So most of the researchers that I've talked to are aware of this, and it's taken very seriously, but it also introduces complexities of how you actually execute a project, how you start it, all of the negotiations and the discussions that need to take place, and all those expectations being met. So, and in particular, sending samples outside of New Zealand adds definitely another magnitude of challenge of meeting those expectations. For example, you need to make sure that the service provider that you're sending samples to, you may well need to make sure that they understand all of the expectations and they respect the where the data goes, where the samples go, how they're handled, how they're returned to the original owners, and all of those types of aspects. It's not trivial to do that. Shipping samples internationally is expensive, and it can compromise your samples. Paul talked a little bit about sample prep at the end, but yeah, it is very important to have good samples, and good samples equals good data. And if you ship them, then the DNA can become a bit degraded or chopped up or get nicks, exposed to UV, etc. All those types of things can compromise the end result. So it is very difficult and cumbersome to send samples internationally. I think people in New Zealand are realizing this. There was some decisions made many years ago that, okay, we think we can just send stuff offshore and sequence it elsewhere. But that is challenging, and it's borne out by the fact that most of the people that are doing PacBio sequencing in New Zealand are people that are connected to international collaborations, and they have gone through a lot of effort to work through the things that are required to send samples offshore. And for those that haven't, often they just default to accessible technology such as Oxford Nanopore, and they compromise. But I guess I would argue that because PacBio HiFi is so good and such a game-changing technology, we really should be thinking about doing some of this in New Zealand. So with that, I would invite you to speak to us about how we can help you. Paul and I and the rest of the Millennium Science team are here to help and advise and see what we can do to get access to PacBio sequencing in New Zealand. So with that, I thank you. And any questions?
Thanks, James. Just waiting to see whether anyone's. I thought that it definitely might stir up some questions. No.
This was a sort of ironic comment almost here. Only seems like, you know, yesterday to me that you know, that the barcode of life projects were happening rather than going for whole genome sort of information. And this is the barcode of life, where you just sequence a single sort of conserved gene or a region of a conserved gene, like cytochrome C oxidase or Rubisco in plants, or ITS region of ribosomal, and that was being done with Sanger. But even that's been sort of adopted now onto PacBio, where they're multiplexing tens of thousands of different samples, looking at that gene and then sequencing it with PacBio as well, which I think is quite ironic that even that sort of Sanger level from years ago is now being converted for PacBio too. That's just an aside, really, but yeah.
I noticed there's a question in the chat from Nikita.
Yeah, Nikita.
Identification of novel transcripts, e.g. non-coding RNA. Absolutely, definitely. My understanding is that most non-coding RNA is polyadenylated. Most of the, not that that's a deal breaker. Most of the protocols with PacBio utilize poly-A tail to produce a full-length cDNA transcript, which you then make a library out of and then sequence. But you can use targeted methods to pull down particular transcripts of interest that you want to study. So yes, absolutely, Nikita, it's a strength of PacBio. I'm happy to talk to you about how we could do that. Would PacBio be used for bisulfite sequencing? That's a good question. The short answer is yes. There is some work that has been done to show that you can modify the conditions of bisulfite conversion of DNA such that you have a more gentle bisulfite conversion, which stops the fragmentation of DNA that typically occurs with bisulfite sequencing. So therefore you can convert, you can do bisulfite conversions and end up with, and still have long molecules and be able, and the advantage of that is that you're able to phase across a whole region. And cytochrome P450 genes is an example of where that's been done before. So you can accurately determine the star alleles as well as determine methylation of those genes. So yes, you can do it. It's a little tricky, and we can help you design that or point you to the resources of how you can do that. The methylation thing, we haven't talked about that at all, but PacBio sequencing does give kinetic data when you produce when you call the sequences or the bases as they're incorporated. They get incorporated at a different rate depending on the methylation or the modification that's on the DNA strands, and you can see that in the kinetic data, and you can model it, and you can call the methylation. That's relatively trivial for bacterial methylations, so we can do bacterial methylation studies very easily. It comes with the data. For eukaryotic methylation, the signatures are a lot more subtle, and they're more difficult to detect. But you can do it now. And the reason, and this is another, gets back to the game-changing HiFi, right, is that PacBio found that with the HiFi sequencing, you get so many passes across one molecule that you get enough data to be able to give a probabilistic determination of whether there's a methylation there or not. So PacBio are actually working on neural networks to call eukaryotic methylation in the HiFi data, and they've got some interesting results showing that they get 85% precision and recall of methylation in eukaryotic whole genome sequencing projects.
Yeah, it's really advantageous that you're getting data from both strands as well. So not only you multiple passing, but you're getting positive and negative strand, and often these are in little palindromes, so you actually see those sites. And yeah, it's very useful in that regard. Yeah.
Great questions, thank you. Yeah, Ron Masons, thank you. And we're actually going to give one little prize away now, one Prezi card, and then one at the end of the 10X as well. So we have Kim Parker. Your name's popped up on the random prize. So yeah, congrats, you got a $50 Prezi card, and we'll be in touch about that. And I am now going to pass back to Fraser. He's going to chair the 10X session. And thank you so much, Paul and James.
Thanks, Lena. Yes, we are down to the home stretch for our final two talks of the day. And this is a session that I've been most looking forward to just personally. I'll just begin by saying, you know, we're very lucky at Millennium Science to work so closely with our suppliers, and 10X is a great example of this. Our next speaker, you know, many of you might be familiar with if you've worked with us on 10X projects or you've entered some of our fellowships. It is the Science and Technology Advisor for ANZ, Dr. Yvonne Peterman. She's an absolute whiz with all things 10X, and she is here to share some technical knowledge on the Visium product, which I personally love. So on to you, Yvonne.
Thanks so much, Fraser, for this kind introduction. You know, we love working with Millennium Science. I think it's a really great and tight working relationship, even though we are not on the ground in New Zealand, unfortunately. I think we try our best to come across and support sort of over the ditch, really. So thank you again. And just with this, I want to use my next sort of 20 minutes to introduce the Visium spatial gene expression workflow, or the product as such, with a few updates of what's coming, as well as, you know, giving you some examples of use cases outside of cancer, which will be followed by, after my talk, you'll get a great example of Visium and how it can be used. So it's the number of publications that we are seeing from scientists utilizing Visium is really rising and increasing. The majority of those is still on Visium fresh frozen. We have launched Visium for formalin fixed paraffin and embedded tissue this year. So we are now seeing also those results coming up in the preprints. So some really exciting data in all fields of science, really. We are also venturing into plant with this product, and I have a couple of examples to show as well in today's presentation. And this here really is a slide that I personally really like showing, because it does highlight the power of spatial transcriptomics in the context of cancer or of tumor treatment, specifically with tumor infiltrating lymphocytes, for example, where here the reactive T cells would have to penetrate the tumor to eradicate the cancerous tissue cells. And in order to actually track whether that occurs or not, a Visium is actually a really powerful tool to look at this. So here on the top, we are actually seeing a tumor stained in hematoxylin eosine on the left, and on the right we are seeing the positive tumor infiltrating lymphocytes, and you can see that they are infiltrating the tumor, which does mean a good prognosis for this particular patient. Whereas at the bottom you can see in the same staining on a different patient, that is not the case. The T cells are not infiltrating the tumor, which means in most cases prediction of a poor therapy outcome and poor prognosis. Briefly wanted to highlight the slight differences between the two sample types that we've enabled now. So for FFPE tissue, for the formalin-fixed paraffin-embedded tissue, we are utilizing large probe sets that we have designed to target protein coding genes. We have targeted about 18,000 genes in human and 20,000 genes in mouse. So these are the two species that Visium FFPE is enabled for at the moment. And Visium fresh frozen is a species agnostic. So here we are capturing mRNA, polyadenylated RNA, directly on our Visium slides. And here we also have lots of tissues that we have tested already being compatible between the two methods. So if you are interested in venturing into this technology and you have a specific tissue type that you want to know works, please get in touch with us or check out the website. There's a comprehensive list of everything that's been tested and we know it performs on the slides. A really important thing for our customers is that the data analysis side of things is sort of a fairly easy turnkey type of analysis, streamlined, and comes with the technology itself. So you can download both the Space Ranger and the Loop browser for free and explore your data. Here is shown the visualization tool, the Loop browser tool, where you can actually pick each of these barcoded spots on the slides that you want to integrate and look at in your specific tissue area. And what these spots mean, that will become clear also very shortly. But sort of the key importance of the Visium technology is that you're really capturing two types of data sets that you can combine. So it's on one side, it's the histological image. You have the choice here with between hematoxylin and eosin staining, or you can also use protein staining, so immunofluorescence. So you can pick either of them, but you have your histology image. And on the other hand, we are capturing the whole transcriptome within the section. And how this is portrayed here in one of those typical T-SNE plots, each spot here doesn't represent a single cell. Each spot here represents the transcription profile of a barcoded spot. And depending on your tissue type, that can be anything from 2 up to 20 cells. And the power of this technology is, of course, that you overlay these two. So you can not only see the gene expression as a standalone, you can see clustering of your data in specific cell types, but you can then change the view and overlay this gene expression pattern with your histological image, and then pick these barcoded spots or these areas of interest to analyze. And so this is really a technology for true discovery. So if you have no understanding of your specific sample type or disease type, you take the entire section and you can discover the gene expression profile across that entire section without having to pre-select any areas of interest. So you cover and analyze the entire section, which is visualized here with all these different barcoded spots overlaid. So you can see the total unique molecular identifier counts as well as the total gene counts in this particular sample here. And what this allows you to do is to assess the disease complexity. You can look at new biomarkers. It allows you to identify new cell types. You can map the spatial organization of cell atlases and also look at spatial temporal gene expression patterns if you want to. So we have examples across all those different sort of types of applications. So really the power is that you are looking at about 5,000 of these spots covering, or that your tissue covers. Another interesting capability of this technology is that you can co-detect protein as well as the whole transcriptome on the identical same slide or section that you have applied on the slide. Currently, we have the capability to do this with up to 6 proteins via antibodies. You detect them via immunofluorescence. In this case here, we have IBA and AIF1, as well as vimentin, which distinguishes mesenchymal tumors from endothelial tumors, and DAPI is usually used as a morphological stain. And you know, you can overlay this again in the tissue view. But what's coming very shortly, so in the early phases of 2022, is the capability to highly multiplex and co-detect protein and whole transcriptome on the same section. So we will increase the capability, you know, from starting as little as tens to hundreds of proteins that you can detect simultaneously. And this is going to be done via oligos that are hybridized to the antibodies, and we are basically capturing just that little information from the oligo rather than measuring immunofluorescence via the antibody. So that allows us this high multiplexing in the future. And this adds another layer to the data sets that you can capture with Visium. So you get your morphology. Here again, an example of H&E staining. You get your gene expression cluster, but now you are getting your protein expression as a separate cluster. And again, I want to highlight this is all done on the same section. So you don't need to computationally merge a different section where you do these assays on individually. It's captured on the same section. We will come out with an immune oncology panel to start with that will have about 45 to 50 antibodies in there. And typically we have these immune markers. They are quite lowly expressed, post-transcriptionally or post-translationally regulated. So actually having that capability of looking at this in the same section paired with the expression profiling will really add in a lot of information to analyzing this set of samples. And here is just a snapshot of a few of these markers, but as I said, this panel actually has over 45 protein markers in them. And as we go and after launching the first panel, many more panels will be coming, and you will also be able to generate custom panels going forward. So how does it work? I'm sure a lot of you would have seen this slide over and over. The key to this technology really is you don't actually need a specific instrument to perform this. The real magic happens on the Visium slide. It has 4 capture areas. The dimensions are 6.5 by 6.5 millimeters. So we do realize that it does pose some challenges depending on the tissue types and organs that you're looking at. But within these capture areas, we have about 5,000 barcoded spots evenly distributed and hybridized onto the glass surface. And each spot has a diameter of 55 micrometers. And that really is what defines the current resolution of this platform, which is again, depending on your tissue type, anything from 2 to 5 up to 20 cells within one of these spots. And similarly to our single cell solution, the oligos look very similar in terms of their structure. The difference is that they carry a spatial barcode, which will map each of the captured transcripts via the poly-DT tail back to its spatial orientation or spatial position on the slide. The workflow itself is once you've optimized sample preparation, sectioning, staining, etc., once that's all optimized, actually going through with the entire workflow up until sequencing ready libraries, it is quite fast. So it can be done in 1 day for fresh frozen tissues. It will take 2 days for the Visium FFPE section, because we do incubate the probe set that I mentioned to you that we have for these tissue types overnight. So that really, just because of that long incubation step, takes it out to 2 days. So you will need histology labs, or you will have to have capability to produce these tissue sections, depending on, fresh frozen or FFP, it will be a cryotome or a microtome. You need imaging capabilities, and you need a service provider or your own Illumina sequencer to sequence these Illumina compatible libraries once they've been created. So as I mentioned before, there is a few differences in the workflow. The key difference being that we need a deparaffинization step in the FFPE workflow, and our decross-linking and overnight probe hybridization step, they are unique to the FFPE workflow. The other workflow steps are fairly similar. Permeabilization and barcoding, as well as the actual library preparation construction, also vary slightly, and that is due to the different nature of probes and transcripts that we are capturing. But what's the same again at the end is you have Illumina ready, Illumina compatible sequencing ready libraries. And I mentioned this before, we are actually using the same Visium slide, but capturing is done slightly differently. So fresh frozen tissue is being permalyzed once applied to the slide, and then we're capturing the mRNA from the tissue directly on the slide, and then we perform cDNA synthesis. And in FFPE tissue, because of the nature of the cross-linking and the denaturation that occurs with RNA, we're actually not capturing mRNA directly. The probes hybridize to their targets, and one part of the probe actually has a synthetic poly A tail attached to it. So once they bound to their targets, we ligate them, and then we release them onto the slide. They get captured there, and then we perform probe extension. So just a few different steps in there. We've put in some comparison here between the two workflows. If you want to get into these techniques, there is a few experimental considerations and really important things you shouldn't skip. You absolutely welcome to always contact us. We are more than happy to talk you through them, and with our FAS guide you through some of these processes. But very important is to assess RNA quality within these sections, and it's done with the RIN value on fresh frozen. And FFPE, we found that the DV200 value is a lot more predictive of the outcome of this assay than RIN. And DV200 is the percentage of RNA that is longer than 200 nucleotides. And we've chosen this because the probes have a certain length and bind to, they need a minimum transcript length to actually bind successfully. We recommend practicing sectioning on standard glass slides with just drawing out these capture areas. We have a template in our user guide, and you can practice as long as it takes until you feel comfortable with this process. In the fresh frozen workflow, there is actually a slide reset protocol available. So if it does go wrong, once it is hybridized to the final slide, it can be removed. However, that is not yet available for the FFPE workflow. So the practice is the best in that setting. We have removed the tissue adhesion test slide as a necessity up front, but please follow the best practices for tissue adhesion in this workflow. And for fresh frozen, there's a few guidelines around sample storage and sample embedding as well. And lastly, the last few minutes, I wanted to use to showcase some applications of where Visium has really shown its power already, and some of these are already pre-published, and some of these have been generated in-house. So here is an example of ovarian cancer, where currently, with the capability of labeling up to 6 proteins, the researchers here have used pan-CK as an epithelial marker that's FIT-C labeled, and then we have CD45.
First, can you hear me? I don't.
Thank you. And then we have CD45 as a pan-leukocyte marker, which is commonly used to mark the stroma. And so you can clearly see here that it's you can distinguish these compartments very clearly by using these two antibodies with the immunofluorescence workflow. But then when you look at the gene expression of the same section, you can actually see there is great heterogeneity in expression to be seen. So you can look at the different clusters that have been identified based on marker gene expression. And you can now zoom into each and every single cluster that's been identified. For example, if you're looking here at cluster 1, you can now look at the top expressed genes, for example, but you can also look at the least expressed genes, so that's completely up to you. We've chosen here the top expressed genes, and within those, you can look at any of the genes, so top 2A or XPO1, and you can see here their expression pattern across the tissue section. So really quite powerful to have that extra expression information of the same slide. And very similarly, you can do this with B-cell markers that are detected in the stromal compartment, where you look at these specific targets, you look at the gene expression pattern, and then you can look at specific B-cell markers within the section and look at the gene expression levels there. So I've picked neuroscience as another example because we often get the question, well, your capture areas seem to be quite small. My tissue that I'm interested in is a lot bigger than that. What do I do? So this is an example of human prefrontal cortex analysis, where the authors here picked a specific area and then chose 6 different cortical layers where they performed sectioning and placed it onto these slides. So there is a method available that shows you how to score a tissue block to make it fit on our specific capture area, to really then allow you to analyze your tissue, and later on you can put that information back together bioinformatically. So here they reveal utilizing specific gray and white matter marker genes, and then they look at they were identifying specifically gene expression patterns that were enriched in each of the different layers. So that was really quite interesting. They went through each layer and shown very specific, very distinct layer enriched gene expression. And another powerful thing that this group showcased is adding single cell or single nuclei data, in this case single nuclear RNA sequencing data from the very same sample types, map this across the different layers that they have analyzed also spatially, and then were able to showcase specific spatial registration of these single nuclei RNA sequencing in autism, in the autism spectrum disorder donors. So if you want to find out more, I can provide the DOIs for these publications. And lastly, what I find specifically very exciting is the fact that scientists make this work on plant. It is not a supported 10x workflow at this point, and that only means we have not tested this in-house and validated the protocols, but we are now seeing it actually works really quite well for specific plants. We've seen this work in seeds as well, so it really opens up some exciting avenues there for plant researchers. And it does take further optimization in the workflow, in the staining and the permeabilization, obviously because plant cells are quite different in their structure than mammalian cells. But I just wanted to showcase here it can be done, and really interesting data from the different sections of a seedling, for example, can be captured. Scientists identify very specific numbers of genes. You see very similar clustering that we see in a typical mammalian cell or tissue Visium experiment, for example. this is really quite exciting to see the outcome of this, and that this works in the plant setting. So they've used 3 replicates here, which I think for plants specifically, it's recommended to do replicates. But the discussion about replicates, how many samples, is something we can have offline with anyone who really wants to get into this. So yeah, Visium's spatial gene expression has a lot of really exciting features. You know, I think the most exciting one, I think, is the co-detection of protein and whole transcriptome, that you don't need an instrument, so you could really perform the histology in your lab and then send the sequencing libraries off to the various sequencing labs in New Zealand. And then, of course, as I mentioned, we are here to support you as much as we can from a distance, and hopefully in person soon. Okay, with that, that's the end of my presentation. Thank you. Don't know if there's any questions.
Thanks, Yvonne. That was a fantastic presentation. And yeah, the plant data especially looked very, very exciting, and yeah, quite sharp imaging there. That was awesome.
Yeah.
I've just put up a poll that people can sort of click on, and these are some of the new technologies coming out from 10x fairly soon. So if you're interested in any of these, give it a click and we'll get in touch with you and set up a meeting with Yvonne to kind of talk about these further at a later date. If you have any questions, please pop up your hand. I think we have time to maybe just do one quick one if there's anyone that has a burning question. Just scrolling through. Yell out if you do have one. Nope, nothing coming through. Is there anything you'd like to comment on, Yvonne, in regards to the pipeline coming up for the spatial platforms?
Yes, exactly. So I'm just seeing this in the poll. Obviously, we have high definition coming out, Visium HD, second half of this year, which will take the resolution of this platform or this technology to single cell or sub-single cellular level, with each of these barcoded spots measuring 4 micrometers. That is not far off, so another 6 months, and this will be on the market. The larger capture area slides are coming out. There will be 2, and they will have 10 centimeters by 10 centimeter capture areas. So if you are working with larger tissue sections, this would be of interest to you. And in terms of an instrumentation, the site assist I see here on the poll, that is actually really exciting when this is going to come out, because it will take the upfront sample prep pain of the workflow a little bit out of your hands. It does it automatically. So you do perform sectioning on a standard glass slide. So really there is no stress involved. And then you load this up on the Visium, on the site assist that has loaded a Visium slide onto it. You pick the area that you're interested in transferring onto the slide. And within the instrument, it basically pairs 2 of your sections that you've put in with a Visium slide, and we actually already capture the RNA transcripts inside that instrument. So there's going to be fluidics in there, there's going to be a microscope in there that also is going to take an image of your paired and captured sections on the Visium slide as well. So this is going to automate that process. It also makes, sharing samples within an institute, for example, a lot easier. So the accessing Visium is going to be a bit easier with site assist. So that's coming out shortly, as well as in situ, although in situ is a bit further away. That's another year, around another year. But that as well is going to be very exciting. So we could probably have a separate seminar on that once we can share more data.
Awesome. Thank you very much, Yvonne. I just saw a question about recordings for the event today. So yes, we will be making the recordings available. And of course, Yvonne's always available for discussions as well. So if you would like to have a one-on-one discussion about your project with Yvonne, just get in touch with Elena and I, and we'll set that up for you. So yeah, thank you very much, Yvonne.
Thanks.
For our final speaker for today, she hails from the University of Otago. She's a second-year PhD student, and I'm personally very impressed with the data that I've seen coming out of this lab. I would like to introduce everyone to Holly Pinkney, who is, I guess, a pioneer in the Visium technology in New Zealand. So I'm I'm really looking forward to this this talk here. So all yours, Holly, take it away.
Cool. Thanks, Fraser. G'day, everyone. I will just share my screen. Just a minute. Ooh.
I think, Yvonne, you might need to stop sharing yours first.
Sorry about that.
It's okay. Cool. All right. Does this look correct to everyone? Is that the right way around?
Just swap the displays.
Oh, okay. Where is that under?
Oh, that's a good question. That's as far as I go with IT.
Yeah, it's a little bit different on this one.
Ollie, I think if you can, do you see the timer in the upper left corner, and then there's the stop, and next to it is another button, like a roundish arrow. Can you click on that?
In the top left corner?
Yep. So where the timer is, can you see the timer?
Oh, yes.
Yep.
Yeah.
Try clicking on there.
That one? That restarted?
Yes, that one.
Did it work?
No.
No.
It should work. It should do the thing.
Perhaps just say use slideshow. Maybe just say click that one.
Where, sorry?
Use slideshow, top left-hand corner. It says end show tips and use slideshow.
Oh, okay. Oh,
yes, there we go. Perfect.
Perfect.
Okay, sorry about that.
That's okay.
Cool. Okay. So I'm Holly. I'm in the DMI lab at the Department of Biochemistry at Otago, and I'm using Visium to identify biomarkers for colorectal cancer. So just a little plug for my Twitter. If that's something that you might like to follow along on, you can jump on there and follow me for that. And also, before I begin, I have a small disclosure that my project does have some commercial interest. So for IP reasons, I can't go into specific details about some of the biomarkers that I'm identifying, but I can walk you through what I've done and how I found my experience using the Visium technology. So I hope you enjoy. Cool. All right. So colorectal cancer globally last year was the third most commonly diagnosed cancer, and was responsible for almost 10% of all cancer deaths. It's also a really big problem in New Zealand, where we have high incidence and mortality rates that disproportionately affect Māori and Pacifica populations. So it is a really big problem here. And most of the mortalities from colorectal cancer are from metastasis to distant organs. So this is where the primary tumour grows, spreads usually via lymph nodes to distant organs. And the 5-year survival rate for that is really concerningly low at just 11.4%. So in terms of colorectal tumours themselves, bulk sequencing takes the average expression of the whole tumour. So that includes multiple subclonal populations that could be within the tumour, and immune infiltration as well. So you're getting just the average expression of everything. But using spatial gives you a much finer resolution image of the tumour microenvironment, because you're capturing just small parts of the tumour, and you can map the expression back onto that. So that's a really exciting thing, and something which I'm really excited to be using. So long non-coding RNAs are what our lab is interested in. They are really highly suited as biomarkers. They are long non-coding RNAs in 200 nucleotides or greater, and they don't encode a protein. They have a lot of regulatory functions in the nucleus or cytoplasm under normal conditions. And really importantly, they are expressed in a tissue and cell type specific fashion. So that makes them really suited as biomarkers. In cancer, their expression is often hijacked. So lincRNAs can be oncogenic drivers or tumor suppressors. And there's lots of examples of these. Oncogenic drivers are usually upregulated, and tumor suppressors are usually downregulated in cancer. Also, lincRNAs have been used in the clinic. Well, one lincRNA. This is the one approved lincRNA in the clinic at the moment, which is the one approved biomarker lincRNA in the clinic, which is PCA3. So that is expressed only in prostate tissue, nowhere else in the body. And it's really lowly expressed under normal conditions, but in prostate cancer, it's really highly expressed. So I guess that shows the potential of lincRNAs as really specific biomarkers for disease. So Ivone has covered this in detail, so I won't go into further detail, but basically my aim is to use Visium to identify lincRNA signatures across tumor and metastatic tissue, and comparing that to normal tissue and identify biomarkers for metastatic colorectal cancer. So the patient tissue that I've been working with has been from the Dunedin colorectal cancer cohort, and that is fresh frozen tissue. So I have two patients. Patient 1 is a male, and he had a rectal tumor. And patient 2 is a female with a sigmoid tumor. So they're both colon cancers. They're slightly different locations of the gut. And patient 1 was earlier stage, so it's stage 2, but had a recurrence into the liver in later years. And patient 2 was stage 4 with an ovarian metastasis as well. So the sections I have collected are 2 tumor sections for both patients and adjacent normal tissue as well. For patient 1, unfortunately, I was unable to section the liver met, so not all the tissue in the colorectal cancer cohort is suitable for use with Visium. Some of it doesn't have high enough quality to be sectioned. But for patient 2, I was able to section the ovary met, so that's really exciting. So patient 1, this is what the tumor on the left and the normal sections look like. So if you're unfamiliar with H&E staining, the darker areas are the more highly nucleated regions, which means they're dividing faster and therefore probably malignant. So that's, I don't know if you can see my mouse, but in this little region here. And up here is kind of some sort of stromal region. For the normal tissue, this is basically a kind of cross-section of the gut. You can see some crypts in here, and this area here is the lumen. So these are archival tissues. You can see they're not in perfect condition, but they have been able to provide me with some really great data. For patient 2, this tumor is a little bit different because it contains some normal tissue as well. So over here you can see some crypts, and then on this side is the more malignant region, and then there's another little bit of malignant area around this side too. So that's really cool that we can see the border of normal into tumor, which is something that's really exciting to be able to look into. And some more sections for patient 2. The normal section here was embedded in the opposite way to patient 1, so you're looking across the crypts there. And patient 2 ovary metastasis looks a little bit messy. What's going on in here is all these dark regions here are metastatic adenocarcinoma of the glands of the ovary, and there's some necrotic regions in the middle there. And all of this light stuff here is desmoplastic stroma. So that's really fibrous and has low cellularity. So that I decided not to focus on that, but just select only the spots that covered the metastatic adenocarcinoma to look at in more detail. So after sectioning everything, I aggregated all of the samples from both patients into one big kind of super pool, and then I performed dimensionality reduction and clustering. So this means that clusters can appear more than once on any given slide. So like, for example, patient 1, a cluster here could also appear in patient 2 if the gene expression profile of the spots was similar enough. So you can see here, there was a total of 14 clusters, but only a small proportion of those was in patient 1 in the tumour tissue, and again, another proportion of that 14 total clusters in the normal tissue there. The orange and the pinky areas in the tumour are the more stromal region, and you can see that corresponds quite well to the histology that I showed you earlier, and then down here, the more malignant region. In terms of normal, this is really cool. We can kind of see the different clusters forming around the edges where there's different cell types. And I'm working with a pathologist to identify specifically what cell types we're looking at, because I'm not a pathologist, so that expertise is really nice. And yeah, this whole project has been really great for, I guess, collaborating with other people who have expertise in the area. So that's been really fun. So for patient 2, you can see the clustering over here for the normal is green and orange. It doesn't look particularly spatially different, but remember the crypts were dotted through the normal section, so that could correspond to that. Alternatively, we might be seeing different ratios of cell types depending on how many cell types the spots are actually capturing. So that could also correspond to different clusters that we're seeing. In terms of the tumour tissue here, we're seeing that kind of a normal looking tissue in teal. I would say it's probably not the same cluster colour as the adjacent normal because it's right next to the tumour. So the adjacent normal tissue is taken right from the ends of the resected colon, which is often quite a big chunk. So it can be quite far away from the primary tumour. And then this time in blue, we can see this is likely a stromal area, and in orange and this kind of yellowy brown colour, the more malignant regions. And what is really cool, I think, is that the cluster from the ovary is primarily this yellowy orange colour, which you can also see in the primary tumour. It's not as much, but it is there. And this suggests to me that possibly this cluster could have seeded the ovary metastasis. So we're really seeing a tumour growth and evolution process here, and that's something that I'd really like to investigate more in my project. So this summary UMAP of the clustering, it has a lot going on. I'm not going to discuss every aspect of it, but essentially down in the bottom left here, we have the normal tissue from patient 1, and then patient 1, the tumour sections over here, and patient 2 clustering up here. So I guess the takeaway points from this is that the patients themselves are clustering out. There's not a lot of similarity between the spots here. So the way the UMAP projection works is that things that are more similar cluster together and things that are less similar cluster further apart. It's kind of similar to a PCA if you're unfamiliar with UMAPs. What I think is interesting is that the normal tissue from patient 1 clustered so far away from the tumour tissue. But again, that could just be from the fact that the section was taken so far away from the primary tumour itself. Yeah, so if you recall, the ovary region from patient 2 was mostly up here. The normal was mostly this greeny colour, so they're pretty far away from each other. So you would think they're quite different. And again, for patient 1, the bluey colours, oh sorry, where did my mouse go? The bluey colours are this kind of more malignant area, and the stromal area was this kind of browny colour. So there's a lot more detail in this that I could talk about, but I won't go into that for now. The next thing that I wanted to talk about are the actual link RNAs that I have identified. These are just some examples. These aren't, I can't really go into details about any more of this, but H19 is a link RNA that I use kind of as a positive control. It's been identified in colorectal cancer before as being upregulated. And as you can see, it has this is log 2-fold change on the scale up here. So it is upregulated across the entirety of the tumor, but not in a particularly specific fashion. So lincRNA CRC1 here is also upregulated slightly more specifically across tumor 1. And what I am really interested in is, you can see here, it somewhat follows the darker, more nucleated malignant epithelial tissue, and it seems to be a lot less in the stromal regions over here. Same thing for lincRNA CRC2. So this lincRNA is obviously much less highly expressed. So once again, that's why the spatial kind of single cell or near single cell resolution is really important, because we're able to capture really lowly abundant transcripts. So this one is expressed much less, but it does seem to have pretty specific expression patterns across the tumor. And once again, we can see it's not in these stromal regions, and it is largely expressed in these malignant epithelial regions. So these are some examples of how lincRNAs can be expressed across the tumor. It is very diverse, but very exciting. And I'm working through a process of screening the potential candidates for both patients at the moment, and narrowing down a list of biomarkers to further, well, biomarker candidates to further validate in the lab. So up next for me is Visium FFPE. So Ivone mentioned that FFPE is probe-based. So that unfortunately rules out me for detecting lincRNAs, because we really require that polyadenylated tail to pull them down. But the Dunedin colorectal cancer cohort has a really big pool of patients which do have tissue stored as FFPE tissue. And we really want to make use of that, because there are 6 patients which I found match every single criteria that I could possibly ask for. So we have normal, primary, nodes, mets, even blood for later validation. So yeah, we're going to test a new unofficial protocol for FFPE tissue that a lab in Queensland has been working on, and they've had success. So I'm going to be trying that up next, and continuing to build, I guess, the number of patients that I have, and get kind of that cross-patient diversity, as well as looking specifically at where lincRNAs are expressed across these tissues. So lastly, my experience with Visium, I just wanted to put this in here to encourage you that it looks complicated, but it's actually a really straightforward protocol, and I've really enjoyed working through it. I had no experience cryosectioning or anything. Maybe you could tell from my sections, I don't know. But I think overall it worked out pretty well, and I was really happy with the results. And when things started to go a little bit belly up, 10X was there really quickly to be able to provide support and ideas of what might have gone wrong and how to improve things. So yeah, it's a really incredible technology. You'll get great results. Tissue quality input is going to equal output, but yeah, overall it's been really amazing, and I'm so excited to continue working with it. So I just like to thank my incredible supervisor Sarah and my lab, and also Dr. Sharon Pearson from the board of the DNCRC cohort. She's also on my PhD committee, and Fran, who is on the ground for the cohort and has helped me to choose patients and tissues and find what suits my project perfectly, and the rest of my committee, and the University of Otago for funding my stipend. So thank you so much.
Awesome. Thank you, Holly. Excellent presentation. And I have to say the sectioning looked excellent. I've done a lot of sectioning in my life, and yeah, that's definitely better than what mine looked like in my first year of my PhD or second year of PhD. So well done there. Yeah, let's open it up for questions. So feel free to use the hand up emoji icon thing to ask a question, or you can pop it in the chat if you're shy. Let me know if I've missed it, if I can't see it as well. I'll just ask you a question, Holly. So, I mean, Yvonne touched on briefly the Visium HD product. Do you think that's going to be something of use to you for your project moving forward? Just because you had mentioned, you know, some of those lowly expressed link RNAs that could be associated with a cell cluster, but do you think it'd be advantageous to kind of pinpoint kind of exactly which cell you'd be interested in looking at?
Yeah, the Visium HD is really interesting because it really does get into that nitty-gritty resolution area, and I think that's really exciting. Yeah, so it'll be interesting to see whether we can go ahead with it. Yeah. Oh, there's a question in the chat. So how deep do you have to sequence to get enough link RNAs to quantify